and what we did last time, please. Yeah, just can you remember me? The first step that we did it. Yeah. Just. Hello? Yeah, please. So put our and now on agar plate. Good. So for what? For the what is the code for this one? And we incubate it. Good. So why we put in this plate? For the first step is the isolator. Good. So separate this two microorganisms that is in mixture. Yeah. So after shaking the plates, we did the please there. Yes. Yes, please. So first we did the isolation. And next we did the please can you participate please? Yeah. The staining. We use a differential staining, that is the Gram stain. Yeah. So this is the first part. For the Gram staining, you can so you can identify or your microorganism. Yeah, you have to describe your bacteria. This two, one it's Gram positive, it's one it's Gram negative. Yeah. So in your slide for the Gram staining, you have to describe the shape and also the arrangement. So if your for example it's like a road, arrangement is like stuff or like a single or a strip. So and for the shake plate, so we finish with the Gram staining. Yeah. So after incubate your plate, you have to interpret or see what's happened for your plate after incubation. Yeah. So what you supposed to see in your plate? So we inoculate how many plates? Incubate. Yeah. Yes, please. Three plates. So the first one is the McCankey. Yeah. Second one is, yeah, please, just to help you, the PA. Good. The third one, it's also the so the blood agar we talk about just right now, and the third one is the neutral agar plate. Good. This is the neutral. So the three plate, and also we talk about the blood agar plates. Yeah, these things. I'm right. Yeah. Okay. So and also, if possible, we can do the mannitol salt agar plates. Okay. So what you think after incubation you get for this plate? What do you think? So for the McKenkie, what do you think you see? You see any growth or not? Yes, please. You see some colonies in your place or not in the McKenkie? What do you think? Yeah, good. So you should get the colonies, the pure colonies, because I said this, the mixture contain gram-positive and gram-negative. Yeah. So the McKenkie is selective which for each for the gram-negative. Good. So for the McKenkie, you get the gram-negative bacteria for your N non-gram-negative. Yeah. And what about the PA? Also, you can get some columns or not? So yes. So here is you have the gram-positive. Good. So just examples to make it clear. So the gram-negative, so the gram-positive is like purple. So it's just make it clear for you. Okay. So if you because it's different than the first practical that I give you, like one microorganism, so it's gram-positive or gram-negative. So if it's gram-positive, so the McKenkie is like the unknown growth. But here you have, I said you have like two microorganisms, one gram-positive and one gram-negative. That's why you get like pure colonies from each plate, the McKenkie and PA that is selected for gram-positive and for gram-negative. Yeah. So you have before doing the biochemical test, you have to describe your colonies. So we talked about this last time. Yeah. Using the different characteristics, how to describe your colonies. Yeah. And for here we can shake the agar plate is better in this step. Okay. So because if we shake the blood agar plate from the beginning, from this mixture, so it's a little bit hard to distinguish between the different type of hemerosis if it's alpha, beta, or gamma. Okay, that's why you can now take a colony, yeah, and take your blood agar plate, and so you can shake your agar plates, okay. And from here also you can shake your agar plate to see if you're gram-positive, which type of the hemolysis is your gram-positive bacteria. Yeah, so that's why it's better to do the blood agar plate after get the pure colonies. Okay, yeah. And for the neutral agar plate, what you can see here for the neutral agar plate, yeah, the growth, good. So we can see what? Growth throughout the medium. Okay, just draw, okay, but you get. So one colony, two microorganisms, what you can get. Yes? So you can get two different microorganisms or two different colonies. Yeah? Yes or no? You have to differentiate between these two microorganisms, depends on the characteristic of the colonies. Yes or no? So just example, just I make it like purple for gram-positive and one red is for gram-negative. You have to so because for the nutrients just allow the growth of the so it's most of the bacteria. Okay, so you can see two type of colonies or two type of the bacteria. Okay, of course it's different, so we talk about this, how to differentiate based on the characteristic of the colony. Yeah, about the shape, size, the color, texture, and for the pigmentation too. Okay, yeah, yes. So here you have to, if it's possible, you have to incubate your agar plates, so the blood agar plates. Okay, so for this is for the blood agar plates. Okay, and also if it's hard to, so if you if so this happen if you are in your in our lab. Okay, it's like supposed to do it. So if you didn't get like a good stain for the gram staining, so it's like if your slide is not clear for you and you didn't differentiate between the two bacteria, the gram positive and gram negative. I'm talking about the gram staining. Okay, you can do your gram staining again. Okay, by taking like a colony and do the gram staining by, so like isolated. So so you have to study, of course, or to describe the morphological characteristic, if you like a cocci or its patella. Okay, you can redo it again for the gram staining if you get a good gram staining from the beginning. So it's okay, you don't have to do it again. Okay, yeah. So that's happen if you like in our lab. Okay, yes please, any question? So to get this pure colonies, you have to follow important step. What is the important step to get like isolated colony from each plate? Yeah, please. The key of the success to get this isolated colony. Yes, please. The aseptic technique. Good. So if you mistake or in the beginning, like you didn't use like good aseptic technique, so you cannot get the pure colony. For example, if you kill your bacteria when using like the inoculating loop, and you didn't let your loop like going down a little bit, you cannot get the isolated colony. So some mistake you can get it. So it's like so for this it's better for you, because sometimes in our lab in this after incubation, some people cannot get nothing in this plate. Okay, so cannot get nothing in this plate, so there are something wrong. Yeah, so that's why you have to use good aseptic technique from the beginning. Yeah, and also maybe you can get like some other colonies in your plate, maybe like a, so for example here just other microorganisms, maybe like a contamination or something like that. So that's why you have to respect the steps for the aseptic technique from the beginning. Okay guys, yeah, so you have a chance for this one, because I will give you all the results. Okay guys, but if you are in the lab, so you have to respect these steps. Yeah, okay. What next? So for now we finished. You describe your colonies, and what you have to do now, please. Yeah, so what is the second part? Yeah, after get your this plate, what you have to do? So the biochemical tests. Good. So how you can do this biochemical test from these two plates? Yes or no? You have to get two sets. Yeah. So we have to do biochemical tests for this gram negative bacteria and one for the gram positive bacteria. That's why you need two sets. Yeah. Yeah. Are you understanding that? Yes or no? Okay, good. So come back to our slide. So this is the first period we did, so we're checking and staining our microorganisms. So the second part is the biochemical activities of this test. What is the different test that you have to do it? So that's why I said you need two sets. So you need two tubes of the TSI, two agar plates, so agar dips, the semi-saturated slant, the urea broth, the nitrate broth, the MRVP broth tube, and the bioescalate. Okay, so this is the different biochemical tests that you have to do it. Okay. Okay. So I think I post in your Blackboard some new material. Yeah. Yes. Yes, please. Do you get it? So there are the 10 microorganisms. So within these 10 microorganisms, there are your two enzymes. Okay? Yeah? Just from this list. Yes? Okay, check your Blackboard, please. If not, I will. Did you get it, guys? Yes. Mahishi, the silver, you get it? The list of the microorganisms. Yeah. I think I posted already. Yes. Do you get it, guys? You said yes, yeah? Okay. So just check your Blackboard and let's see. Okay. So here you have two microorganisms, 1 gram positive and 1 gram negative. You have first to get your test tubes. You have to what you have to do to label your tubes, yeah, for your unknown. For example, it's like, so you have to, for example, just this is for like TSI tube. Okay, you have to label it like from this microorganism that you have in your plate. It's from the PA. So here's the PA plate. So the PA plate is a gram positive. You have to see here. You get, because I will give you like this. So this TSI is like the colonies or the microorganism is from the PA. Okay, and you have of course label by your unknown phone number, like number 5. Okay, for each one. And also here for the McKenkie, you have to take your tube, for example, to TSI. Okay, yeah, and label. So this microorganism is from the McKenkie plate, that is gram negative, yeah, and my unknown is number 5. Okay, for the Macaque, we did not talk about the Macaque. Is like selective and differential. You can tell, so you can see if your microorganism is like what? It's used for to differentiate which what. So it's if your microorganism is like mannitol. Yeah, it's or what's yeah, please. Mannitol or lactose. So it's produce acid, yeah. So it's to differentiate what? If your microorganism is fermenter of what? Which type of the carbohydrate? For lactose, good. So you can also see if you get like pink growth or colony, so it like lactose fermenter. If you didn't get this pink color, so it's not lactose fermenter, like just for the gram negative. Okay. Okay. Okay, so here that's what was I said you have to get two sets. So just we talk about one, and you have to know that you have to do two biochemical from each plate. Yes, guys, just let me know if you understand this thing here, because you will need to set of same media. Okay. Any questions, please? Can you hear me, please? Yeah, sorry. Can you hear me, guys? Yeah? Okay, sorry. So we start, we talk about the TSI test. So for the TSI test, it's used to see how you have to inoculate your tube. So it's like a slant, yeah? How will you inoculate your TSI tube? Yes, please. You can talk in, please, if just make it easy. Question, yeah? For inoculation group, how you inoculate your tube or your media for the TSI? Yeah? Remember or not? You have to shake it and stab. Good. You have to shake in the surface of your slant and stabbing or stab your tube using a needle. Okay. Good. So after this, you have to incubate your plates. After incubation, you see you can get like three things. What is the three information that you get it from your TSI tube? Yeah. First, what you can see here, and how you read your tube. Please, let's go. Just make it fair. And the part of color. Good. You see your slant. Question, if you want to talk, or just mute yourself, please. Okay, so the good, the slant and the butt, so and the H to S, good, to H to S, or the hydrogen sulfide, good. The third things, so the slant and the butt color, so tell us if the type of the carbohydrate that your microorganism can metabolize or not. Yes or no? So because the TSI include like 3 types of the sugar, the glucose, sucrose, and the lactose. So it's depend to the color of your tube for the slant and the bottom. So it's like, if it's, for example, it's like all the tube is like yellow. What mean here? So it's acid, it's AA. What mean? Yeah. Yes. So your microorganism can ferment the 3 type of the sugar. Good. So we have other possibility. If the microorganism can just metabolize or can ferment just the but cannot produce H2S, good. If your microorganism, so there are many different colors. So if your organism do not use any of the sugar, so you can get KK, or the tube is red, because catabolize the protein, and protein produce or increase the pH in your medium. Okay. And also, so here you can tell us about the type of the sugar, of the carbohydrate. Yeah. So it's like glucose or the other sugars or not. Okay, we have many possibilities. Okay, you see here the H2S production. How you can know if your microorganism can produce the H2S or not? How? Please. Yeah, the black precipitation in your medium. Good. So either are black precipitation, must mean H2S positive. If not, there are no black precipitation, means it no H2S. And the third one, yeah, what you can get, yeah, the gas. Good. So also you see if your microorganism can produce or not gas or not. Okay, so it's positive or negative. Okay, I think this for the TSI. Any question about the TSI test? If you need to go back how to read the result for the TSI, it's okay. If not, just let me know. Yes, Anna? No? Okay. The second one, what do you think? We talk about which one? Just let me know here. The second test. Yes, we talk about the SIM citrate. Okay, the SIM. Okay, we talk about the SIM. So here, the SIM, just make it a different color. So for the SIM, it's also for the H2S, yeah. S is for the hydrogen sulfide, yeah. Yes, please. R it's for what? M it's for utility. Good. And sorry. And R is for the indole production or not. Good. So for the same, it's a semi-solid medium. Yeah. If you remember, yeah. So you have to stab your medium using enucleating glue. Enucleating needle. Sorry. So you have to stabbing, and after incubation, you get this results. So what you can get for this the same tip or tube, yeah. So it's like a semi-solid. to study the maturity. So we talk about this. After for the sim, for the TSR, you don't need any reagents. For the sim tube, what you have to do? So you have to need any reagent or not? Yes. So in this your sim tube test, you have to add what? Yes or no? Corvex reagent. Good. So you have to add the Corvex. Corvex reagent in your tube. After adding the Corvex, like some drop, like 3 drops or two or 3 drops, and if there are like a red color, what mean? Indole positive. So if like a layer the top, if you get like, so here is like a red layer in the top if you're of your tube, so mean it's indole positive. So if there are no reds, so your microorganism cannot hydrolyze the amino acid, the tryptophan, and release the indole. So it's like indole negative. Yeah. After adding, of course, the Corvex or indole reagents, and also what you can see here. So you can, and so the indole. So we'll talk about the indole, and the H2S production also is like by black precipitation. Yeah. If you have like get a black precipitation, mean your microorganism like produce the H2S, and also we talk about the motility. So if you're here, like you have many many things. So for example, if it like your tube is like turbid, what mean here? All the tube is turbid. Yeah. What mean? So your microorganism is motile. Good. So your microorganism is motile, is like swim away from the line of inoculation. So you get like turbid tube. Or if the, for example, here is like the turbidity like is restricted in the line of the inoculation, that here is non-motile. Yeah. So here it's like upside, and here is not. And also it's same if you have like H2S production, production. So it's like a black, all the two is like black, and or this restricted in the line of the inoculation. Yeah. So here is the what you get from the sim tube test. We'll talk about the next test. We talk about the simulcitrate test. Yeah. So the simulcitrate test is like blue or green tube. Yeah. Yes, please. It's a green. Good. Citrate. So all this result can help you to identify your microorganism, of course. So for the simulcir, it's a slant. You have to inoculate your tube or your media. After incubation, so if your tube is like turn blue, yeah. So what mean is like citrate positive, because the bacteria so it's turning like blue under a condition. So it's like positive. Yeah. So the most important for this test, it's used to identify of which kind of the bacteria. we talk about it, I think, yeah. The Enterobacteriaceae, yeah. So we talk about the Enterobacter Klebsiella. So this one, it's citrate positive or negative? Yep. I think we talk about this, yeah, before this. Yeah, so it's positive. So it's like citrate positive. And for the E. coli, so E. coli, it's citrate negative. Okay, so the citrate tube or test can differentiate between many types of the Enterobacteriaceae. The Enterobacter and the Klebsiella, it's citrate positive, and the E. coli is negative. Okay. Any question about this three first test, biochemical test, please? Yeah. Any question? No. So we talk now about the fourth test, that is the we talk about the MRVP test, yeah. MRVP. So the MRVP is from the IMVIC test, yeah. So we talk about the R. It's for the indole MRV IMVIC, yeah, yeah. So the methyl red and Voges Proskauer test. We'll talk about the indole. C is for the citrate, and we talk now about the M and the V. For the M, it's for the methyl red, and the V, it's for the Voges Proskauer test, because you have two tests here, yeah. Yes or no? So for this test, it's used to differentiate between also two type of the microorganisms. Which type of the microorganisms that we can differentiate from this test? So it's depend to the end of the product. So if at the end of the product, yeah, after fermentation of your product, yeah. So if the end of product is like, yeah, we have two possibility, yeah. We talk about, just you have to remember, please, which one, the first one is what. So if it's, yeah, the MRVP, please make it fast, very fast. Yellow, brown, and red. Okay, good. So just if the end of the product is like acidic or like a neutral end product, yes or no? So if like you have like acidic end product, so your organism is like organism mixed acid fermentation, fermenter, and acidic fermentation, yeah, and or the end of the product is like neutral. So yes, so the neutral and product, the acid, so acidic and like base, so the acetoin and the butanediol, yeah. So for the acidic, which type of the bacteria? So we talk about this, please, before. Just make a summary or just two. So red positive glucose fermenter, good. So we have here the E. coli, yeah. And for the neutral, which type of the bacteria? Give me one, yeah. the Enterobacter aeruginosa, yeah? Yeah. So for you have to inoculate. So the MRVP test. So it's a broth. You have to inoculate your broth. Yeah. And after incubation, you have to split your tube in two halves. Yeah. So here your MRVP broth. You have to split within two tube. Yeah. So you have to use one for the MR test, and the other one is for the VP test. Yeah. So for the methyl red test, you have to add like some drops or like 3 to 4 drops of the methyl red. If you get like red, so if the tube is like turn red, so it's MR positive. Yeah. Yes or no? And for the VP test, you have adding two type of the reagent, the VPA or the VPA and the VPB, or VP1 and the VP2. It's so two different reagents. Okay. And you have to add like the VP1, okay, and then adding the VP2, and you have to wait how many to get for the reaction or for the biochemical reaction for the VP. How many minutes you have to wait to get the result? Yeah. Like 30 or between 20 to 30 minutes. Okay, you have to wait like 20 to 30 minutes to get the result for the VP test. If it's turned like red, so your microorganism is VP positive, and the end of the product, so it's like neutral. Okay, so the MRVP test differentiates between different type of the Enterobacterium C2, the E. coli that is has a like acidic product, and for the Enterobacter aeruginus has neutral end product. Okay, so it's important that to differentiate between your so to identify like your endpoints. Okay, any question for this, please? Any question, please? Yes, guys? No? Just to make sure, please. So we did the TSI, MRVP, SEMO test, indole. What else? Yeah? The urea. Good. So the urease test. So we talk about the urease test. So for the urease test is like also it's like a broth or like, yeah. So it's broth, yeah. So you have before the inoculated tube is like yellow, okay. After inoculate and incubation of your tube, you can get like two different color, okay, or just one of them each. So what is the first possibility? Yeah. So here you see if your microorganism can metabolize the urea. Yeah. So if your microorganism metabolize the urea like slowly, or yeah, rapidly, or like quickly. Yeah. So if like you have a slow urease reaction, yeah, so the tube is like turn like orange, yeah, yes or not, or like slight color change. If your microorganism metabolize the urea like rapidly or like quickly, so the tube turn like pink good or like bright fisher color, so it's like pink color. So I don't have this color, so it's like different than the first, like pink. Okay, just I don't have pink color. Okay, so here it's like slow, so the organism metabolizes the urea slowly, and here is like quickly or like quickly. Yeah. Sorry. So we talk about the urease. What next, please? So the tubes. So we talk about the other test we didn't talk about is the catalase and the oxidase. So it's important test. Yeah. Yes. Any test we missed? So we miss anything? So, yeah, we did talk about the nitrates. Thank you. So we did not talk about the nitrate. Yeah. So before we talk about the catalase and the oxidase, we talk about the nitrate. Yeah. Thank you. For the nitrate, you see if your microorganism can. So what we see for this one, for the nitrate, we talk us about the nitrate. Yeah. Yeah. Yes, please. Yeah. For the nitrate, just can someone explain us about the nitrate test, about the introduction of the nitrate? Yes, please. Who is a volunteer for us, please? Yeah. Just two. Yeah. We'll talk about the nitrate, please. Mahishi, yeah. So we see the ability of the organism can reduce the nitrate in the nitrite. So here is the reduction is complete or incomplete. Yeah. Just give me your answers, please. So your turn, please. So we talked about this before. Just give me what you know about the nitrate reduction. Yes. So if the microorganism reduce the nitrate to nitrite, so it's like incomplete reduction. So if the microorganism reduce the nitrate to nitrogen gas, so it's a complete reduction. So all the nitrate reduces to gas. Okay? How much? To nitrogen gas. Good. So if no red color, so the nitrate didn't reduce or reduces to nitrogen gas. So first, after incubate and incubate your tube, yeah. So you have to add some reagents, yeah. You have to add the nitrate or the nitrate reagent A and B, yeah. Yes, guys. So after adding this reagent, if your tube turn like red, mean the nitrate reduces to nitrite. Yes or no? Yes, please. So here we have to adding the reagents, the nitrate. A and B. If the tube turn red, so it's like positive. Not sure of this. Good. I'm using some enzyme. Thank you, Silva. To remember about the enzyme. So the microorganisms use the nitrate reductase here, the first step. Yeah. The nitrate reductase. So the microorganism can produce the nitrate reductase, okay, this enzyme, to reduce the nitrate to nitrite, okay. So the tube turn like red. And the other possibility, after adding the reagent, maybe you can get like, so not change in the color, yeah, so it's like clear, yeah, yes. So what mean here? So here we get the result. It's like positive. So the microorganism reduce the. It's like so reduce the nitrate to nitrite. Yeah. Yes or no? Yes. Because if the tube turn red, so the nitrite with I, so it's present. Okay, because it's react with the reagents A and B and make this red color. So here it's like here is the presence of the nitrite. Okay. For the other possibility, if you get like ink clear or no change in the color for your tube, you have what you have to do here. So you have two possibilities. If your microorganism, so it didn't reduce the nitrates, or he's reduced it completely to gas. So you have to add the zinc. Good. After adding the zinc, and also adding the HCl, so making the acidic condition. So after adding the zinc and also the HCl, the zinc under acidic condition, so you can get two possibilities also. If your tube like, what is the two possibility here, please? After adding zinc, so if the tube turn, what you said here, yeah, red, turn like red or pink, good, and or it's like still like colors, yeah, or clear. So what's mean if it's like turn red? So it's negative. Good. So so it's negative. Mean the nitrate wasn't reduced, it is still here in your tube, yeah, because after adding the zinc, so the zinc converts the nitrate, yeah, that is present is in the nitrate, because the tube that you know contain the nitrate, okay, and also by that you know your tube have already the reagents A and B, yeah, so turn like red, okay, yes. So that just wasn't reduced, good. Because why is turned red? Because the nitrate that is present in the tube, the zinc will catalyze the conversion to nitrite and make this red color in the presence, of course, of the reagent A and B. So if the tube like still like colors, what you mean here, yeah? So it's positive or negative? Yeah. So it's positive. What mean it's it's positive. Yeah, good. So the nitrate it reduced into gas. Good. So the nitrate here. So just this. So the nitrate reduce it completely to the gas. Okay, you see the difference here. Okay, guys. So here you can see the reduction of the nitrate if it's completely or incompletely completely to the gas using two types of the enzyme, the nitrate reductase and the nitrite nitrite, okay, with I. Okay, guys, yeah. Any question for this piece? So because it's why it's like colors, because the nitrogen gas we cannot detect it by the reagents. Okay, that's why it's like a colorless. So the gas is like so it's like a colorless or like clear. So we cannot detect it by using the reagent, but the nitrite that we can detect it using the reagent A and B. Okay, any questions, guys, about this different type of the biochemical test? Yeah. Yes, please. So we talk about two important tests also, about the catalase and the oxidase. Yeah, so it's help you to differentiate between. So we talk about first about the catalase. So we talk about the catalase. So just to remember you also. So the catalase test. So why we use the catalase test first to differentiate which type of the microorganism. So we'll see. So anaerob from the aerob. Good. So if your microorganism can produce this catalase or this enzyme that can neutralize the toxic form of the oxygen, so that is the type of the aerobes, yeah, or the facultative aerob or the aerotolerance microorganism. If for the aerobes, so it's catalase positive or negative? Yeah, for the aerob, yeah, so it's positive. And for the anaerobes, cannot support the presence of the oxygen. So the oxygen is like kill this microorganism. It's like harmful. So because this microorganism cannot produce this type of the enzyme to neutralize the toxic form of the oxygen after, of course, the metabolism. Okay, so here is like anaerobe and anaerobes and aerobes microorganisms. Good. And also the catalase test used to differentiate which type of the bacteria we talk about just before, and I give you like a question in your test or in your exam. Yeah. So to differentiate what? Yeah. Yes, please. The Staphylococcus and the Streptococcus. Good. So differentiate so both of them is like a gram-positive cocci, yeah? Gram-positive cocci, yes or no? So it's gram-positive cocci. So the Staphylococcus, so it's like pathogenic or not pathogenic, like always epidermis and coccus. So it's so the Staphylococcus and the Micrococcus too. Okay, so this one is catalase positive or negative? Yeah. So that is catalase positive, yeah? Yes or no? Is positive. So differentiates between the two different type of the gram-positive bacteria, that is staph, and that you said Silva is the Streptococcus, that is so the Streptococcus. Or we have the Enterococcus. So we have this type of the bacteria. Okay, Enterococcus. Okay, that is catalase. So I don't know why some people is going out and on or something like that, please. So it's like catalase negative, yes or no? So we talk about the Streptococcus in our maybe this week, okay? The different type of the Streptococcus, okay? We have the Enterococcus, okay? So for this is like catalase negative. So the catalase test can differentiate between the gram-positive cocci and also adults also. So the Enterococcus, we can use which test we didn't talk about the bioesculin, yeah. So we use the yeah. Yes or no? We talk about the bioesculin that can differentiate the Enterococcus, yeah. So the bioesculin for this Enterococcus is like positive. Yes or no? Yes, please. So we use the bioesculin test to differentiate the different type of the Streptococcus, the Enterococcus. So that is bioesculin positive. So the catalase test differentiates between gram-positive cocci, that is the staph, and the Micrococcus is like positive, and also we have the other type of, so we talk about the cocci. What is the other type of the microorganism? The gram-positive, yeah. Just the we have the cocci and the shape, the other shape, the bacilli, good, or the rod. So also the rod is like catalase positive. So catalase positive, it's like for the gram-positive rod, and for the gram-positive cocci, staph, and the Micrococcus. So the catalase test, it's like a important test to add to differentiate between this type of the microorganisms. So it's important to for this in this second period of your biochemical test, okay? Because from your slide from the gram staining, if you get like a rod, so if the catalyst positive, so if the catalyst positive is like a bacillus, so sorry for this, I didn't say it to you. So it's like Bacillus subtilis, that is a rod, yeah. So you have this microorganism also in your list. Okay, if it's like cocci, we see if it's positive and negative. You can differentiate between staph and the strep. Okay, you see here. So that's make it easy for you to to eliminate some microorganisms from the others. Yes or no? Yes, please. And also, of course, by using the other media of test too, that we talk about the MRVP, that differentiates between the E. coli and the Enterobacter aerogenes, and other tests too. Okay, any question about the catalyst? Because the catalyst is important. Any question, please? So make it easy for you to know your microorganism. Okay, guys? So I post already some shots for the catalase and the oxidase, yeah, in your blackboard. Yeah, guide, yeah, yes. Okay, good. So for the oxidase, so for the oxidase test, I think it's the last one here. The So for the oxidase test is used to distinguish the gram negative. So we talk for the catalyst, we talk about the gram positive rod. So for the oxidase test, it's talk about the gram negative rods. Okay, so that's why the gram staining, it's important to know if your NO, the shape of your NO, if it's like a rod, it's like cocci or something else. Okay, so it's important to help you to identify or name your microorganisms, because for this this practical exam in your lab report, you have to give me the name of 2 microorganisms. Okay, if it's like E. coli and staph or something else. Okay, so the oxidase distinguish between gram negative rods or bacilli. We have the important one that is oxidase positive, D. Yes, please, D. That's make like green pigments. What is the name of this microorganism? D. Yes. Yes, the Pseudomonas, good. The Pseudomonas aerogenes. So it's oxidase positive. So this member of the enterics, so which are facultative anaerobe, the other type of the microorganism, are oxidase negative, okay, and cannot use the cytochrome, the cytochrome C oxidase, that is oxidase negative. Okay, guys, I think we talk about all the biochemical tests. Yeah, just make sure you miss something or not. Yes. Any questions, please? So what do you think? We missed something or no? Talk about the... Any questions, please? So for the lab reports due, so I give you like 10 days, okay? So when will be our final exam, please? So I will get sent to you by today your announcement and the result of all this biochemical tests, and also for the sheet plate thickness for the streak the plates. Okay, so by the 6th. Because you pause for today, if you are in our labs, okay, you have to incubate all this test, and by Wednesday you get the result. Okay, so by Wednesday, if you have any questions about your lab report, we talk about so for the lab report is same thing like the first one. So you have like 4 steps, the introduction, or 5. Yeah, what is the steps for the lab report? Yeah, please. Yes. Introduction, body result, and discussion. Also so for the body you have the method and the material, yeah, and you have the result, and you have to discuss the result, and also for the final one you have to do your what else? What is the name for the last one? The conclusion. Good. And the reference, yeah, don't forget name. So you have to, for the conclusion, you give me the name of your 2 microorganisms or 2 bacteria. Okay, good. So you have to use same so it's same steps like the first one. For just I think some of you, I give it to you like some, you have to read the comment that I give it to in the first practical, yeah. So so don't make same mistake for the practical one. Just please check the comment that I give it to you, some of you, okay? If you have any question about the lab report, we can talk about just by Wednesday, okay? Just please, when I give you your plates and your biochemical test, you have to read all the information that I posted to you in your Blackboard. So for any question, we can talk about it just by Wednesday, yeah, our next meeting. So it's the last part for the practical, and we have to do we start about talking about the pathogenic microorganism for different system, okay? Talk about the skin, about the gastrointestinal, about other respiratory and the urinary system too. Okay, so we start our labs again by Wednesday, and we can just talk like between 15 to 20 minutes in the beginning for the. So if you have any question about the lab report or something else, okay? Yeah. Yes, guys. Just let me know if I missed something, or just can you remember me for something else if I didn't talk about it. Yeah. Yes, guys. Any question, or you can talk just yeah. Yes, guys? Any questions or some suggestions or something? Okay. So for the due, so if you have like our final exam is gonna be like 16, so it's gonna be like Wednesday. So I just give you like 10 days. I think it's enough or more. Okay? Yeah, because our last meeting is gonna be Monday 14. Yeah. Yes. So and the exam is gonna be so Monday 14 and exam is gonna be 16. So I think like 10 or 11 days is it's enough, yeah, to do your lab report. There are no more lab reports. Just you have to work in your practical exam for the lab report for your practical exam too. Okay, yeah, good. So make sure that your the last assignment is about for the lab 18 or just for the lab report 18. Okay, yeah. So for the effect of the temperature and the pH, and there will be no more lab report, just the for the lab report for your practical. Okay, guys. Yeah. So 10 days it's enough for you. Yeah. Good. Yes, please. Good. Okay. So your announce I will is same like before. You have to check your email. Okay. And will send you the name, so the number of your NL. So it's each of you get a number, and also the results. Okay, guys. So if you have any questions, just so at least by tomorrow or by tonight. Okay, just I have to verify some results to make sure that I didn't miss something, and I will send it to you. Okay, guys. Okay. Okay, guys. So just I get some messages from some students. Yanis, are you here?
Mama.
Okay, just I'm here like 5 or 10 minutes. If you have any questions, just let me know about your grade or something else. Okay. Okay, guys. Okay, have a nice day. See you Wednesday. Yeah. And stay safe and take care, guys. Okay. Thank you.
Bye, Professor.
Bye. Thank you, guys. So I'm here just if you have anything, just I'm here like 5 or 10 minutes.